So Zain, thanks a lot more for your introduction and for showing everyone my most photogenic side. I'm going to be focusing on what I've participated in without this larger SALMONID collaboration, and I've been mostly focusing on clustering and analyzing the transcriptome dataset. So I'm going to show you a bit of those results. But first, as Mo was saying before, I'll just go quickly through this because we've probably already all heard about it, but Atlantic salmon and rainbow trout share an ancestral whole genome duplication. This occurred around 100 million years ago, and the salmonids started diverging around 50 million years ago. So there is a period where the divergence was within a single lineage, and then after that, there were different lineages that were rediplodizing separately. And Atlantic salmon and rainbow trout further, that their ancestors diverged around 25 million years ago, so there's been further lineage-specific rediplodization within the Salmo and Oncorhynchus genuses for around 25 million years. So the point of this study was partly to try and understand better how the duplicates generated after this whole genome duplication have diverged following this process of rediplodization. So the transcriptomic datasets generated as part of this project were separated into two. First, we have the embryonic datasets, which were part of the DevMaps package. And this was comprised of a total of 14 embryonic stages with a total of 84 RNA libraries. And then for the tissues, this encompasses both salmon and trout. And then for the tissues, same for both salmon and trout, there's a total of 96 RNA libraries that are spread across 8 tissues. So now we can see some of the results of that. So here, these are the embryo maps. So the data, the RNA-seq data, was first we obtained TPM data from NF-core, and this was further normalized. So what we're seeing here is scaled TPM. And some clustering was applied to obtain distinct clusters of expression throughout embryogenesis, which then we further visualized using heat maps. So the different slices you see here, each of these is one SOM cluster that you can also see over here in the line plots. And then we further visualized it using UMaps. So in these UMaps that you're seeing here, each dot is a single gene, and it's placed in this two-dimensional frame depending on its expression pattern throughout embryogenesis. So what this reveals is really the dynamic nature of embryogenesis, where we have some groups of genes that are present very early on, but then very quickly degrade. And then these are potentially maternal transcripts, although it's difficult to say with our experimental setup to completely confirm whether or not these are the zygotic genome is already activated during this period or not. But one also very interesting thing when we have salmon and trout data together is how strikingly similar their transcriptomes are throughout all of embryogenesis. So we have almost one-to-one relationships for different SOM clusters that are really a great tool for comparative analysis. And also it's interesting how the UMAP recreates this trajectory of development, where we have here what are constitutive classes. These have been confirmed to be constitutive in nature through gene ontology enrichment. And they are present towards the middle of the UMAP, and then over here we have early expression genes, and as we go over here we have genes expressed later on in development, and the shape of the UMAP is also quite similar across both species. So when it comes to the polymaps datasets, it's also, as you can see, quite similar across Atlantic salmon and rainbow trout. There are some small differences in the case of the testes, but mostly this is due to a different number of libraries between Atlantic salmon and rainbow trout. And the different categories that were obtained through the SOM clustering can be divided into tissue-specific. So here we have like ova, brain genes, testis genes, muscle genes, liver, head, kidney, gill, and intestine. And then over here, we have classes that are not constitutive per se, but are expressed in multiple tissues. And it's interesting, for example, classes that are expressed in both head, kidney, and liver, that we can suppose that they probably have some sort of immune function, since both of those organs have an immune function. The UMAPs are a great tool to visualize this body maps data, and they, again, place the constitutive genes spot on in the center, and then each tissue, instead of, like before in the def maps, we have a trajectory of development with genes turning on through development. In the body maps, we have a bit of a different scenario where the different tissues extend outwards and create their own specific cluster that it's been colored by some, but the UMAP doesn't really know which gene belongs to what some. It just has its pattern of expression throughout development. But they are very good at separating the different somes together, which tells us that both SOM and UMAP are working well together. And again, this multi-expression class is observed in between each class. So if this is expressed, for example, in between liver and ova, something will probably be present here. So the next thing we did was try to, and this is fresh out of the oven, by the way, do some gene ontology enrichment to better understand the function of each cluster. So for the def maps, I have individual clusters, although I did remove constitutive to try and make it a bit less cluttered. And then, on the other hand, for the body maps, I collapsed the SOM clusters into tissue-specific expression. And then further, I also added constitutive class. I've still not had a chance to have an in-depth look at the multi-expression classes. So we can now have a look at that. I have edited the plots a little bit because each of these is a single term, so it would have been too much of a mess to show here. So I've just curated it a little bit to show what is happening in each rough stage of development. So here we have early embryogenesis, and as we go down, we have terms associated with later development. So we can see that the earlier stages are characterized by the negative regulation of DNA transcription and things like cell cycle. We also have post-translational regulation of RNA and cytoskeleton organization. So all of this tells us that it is likely that these clusters are composed, at least in part, by maternal transcripts. The fact that there is a negative regulation of DNA transcription tells us that even if the genome is likely starting to activate, it's probably largely still being negatively regulated and inactive. And most of the terms associated with this cluster are associated with things like the cell cycle and the structure of the cell. And also RNA post-translational regulation, which is one of the ways that maternal transcripts can do their thing, basically, so because initially they need to be modified at the right time so that they can perform their function. Later on, as we advance into bastulation, we start getting terms associated with cell secretion, cell growth, and cell recognition, as well as regulation of transcriptome, telling us that it's likely that, at least in this period, the terms associated with actual genome activity start to increase. Also regulation of, sorry, I just said that, yeah, and also mRNA catabolic process, which is an indication that likely the maternal RNAs are being degraded throughout here. These are all suppositions, but I think it's a pretty decent likelihood that that's the case. And it will be really interesting to do further studies to further confident, because there's also indications by other colleagues and collaborators that there might be even earlier activation of the genome. So this is something that is not really known in salmonids exactly when it happens, so it'd be really interesting to do further research to further clarify that. Right, so as we go further down into late blastulation and the start of gastrulation, we have mRNA splicing, nervous system development, which is a bit of a shocker because there's no brain in the embryo there. But this is also associated with endoderm development and things like that. The genes, I've looked at the specific genes, and it's endoderm terms and stuff like that. So it doesn't necessarily mean that there's a brain at that point. Also cell death regulation. And further down, we start getting highly enriched terms for cell signaling, cell adhesion, and tissue development. And all of these things are how the cells regulate things like migration, regulating how much they are adhered to each other to be able to move and form the different layers of the embryo, as well as tissue development, which again makes sense for gastrulation. And further on, as we advance into segmentation, we get things like WNT signaling, organogenesis, muscle development, anterior-posterior patterning. Again, we can see the anterior-posterior patterning here. And tube development. So these are all consistent with expectations for these stages. And finally, for the 8th stage, which when the circulatory system around the yolk is developing, etc., we're getting terms associated with circulatory system development, renal development, and also increased signs of increased metabolism, like ATP metabolism and carbohydrate metabolism, and also lots of brain activity and eye development. As we go on into the body maps for co-enrichment, it mostly makes quite a lot of sense for the different tissues. So we have the tissues down here. So we have ovary, brain, testis, muscle, liver, head, kidney, gill, intestine, and then a constitutive class. And for ovary, it's interesting to note that a lot of these terms are found also in the earlier stage, again suggesting that this could be at least partly maternally deposited, I mean. Then going into the brain, well, makes sense, nervous system development, potassium ion transport, cell signaling, and locomotion. Going into testis, we have flagellum assembly, embryo development, things that in general make a lot of sense. For muscle, we've got a big component associated with metabolism, but also, of course, muscle structure development. And then going into, there's a big class here of several terms that suggest that the functionality of these organs, such as the liver, head, kidney, and gill, liver and head, kidney, are slightly overlapping, and they both have, although the liver takes more of a metabolic role, they both have an immune component. So we find terms associated with immune in both tissues. And finally, for gill, we have terms associated with gas transport and phagocytosis, if I'm reading that correctly. And then the constitutive class is highly enriched for things like histone modification, non-coding RNA metabolic processes, and nucleic acid catabolic process. So things that, again, make sense for constitutive. And also there's stuff like cell cycle and stuff like that in there. So all in all, this is providing us with a very good picture of what these SOM classes that we're seeing before see here. So we can point at specific SOM clusters and say what is going on there. And this wasn't necessarily easy to make because for those of you that have not done gene ontology enrichment before, one of the issues we face is that there's a lot of repeated terms due to parent-child relationships between gene ontology classes. So I had to develop a method to find which of these terms were pretty much the same thing and sum them up so that they can be plotted in a single figure and visualized appropriately. And even then, I still had to edit it for the presentation. But yeah, further from this, leaving this, I'm also interested in the constraints acting through development. So in all of these periods of development, there's different homologs present, duplicates from the whole genome duplication. And we're interested in understanding how different processes going on in development are shaping homolog conservation. So to better understand that, I took an approach that has previously been used by Marletta et al., for example, and many others of using the Jensen-Shannon divergence metric to compare transcriptome similarity. However, I did one thing a little bit different. Previous studies have taken the approach of comparing different species. Here we see zebrafish, medaka, I think Xenopus, and chicken. And we can see that there's a period towards late development when organs are maturing, where there is a very high transcriptome similarity across species. And mirroring this approach, I decided to look within a single species at the duplicates generated through the whole genome duplication and apply this same metric to them. And we can see that the divergence in gene duplicate expression is steadily decreasing towards the late 8th stage. We don't have further stages, but development goes on for quite a bit after that. And the expectation is that divergence would go up as is mirrored by the adult tissues that we have here that have been ordered according to their constraint level. So this is really almost done. So this is the first indication that the same constraints operating through development that are shaping transcription similarity across species are also determining how homologs are being maintained or diverged through development. And Manu, who is speaking next, has some really interesting results regarding how this is being shaped by changes in gene regulation. Thank you very much for listening.
Thank you. Questions? We can have a couple of them.